Hello everyone, we are in the lecture series of overview and integration of cellular metabolism in NPTEL platform. We have started the metabolism of amino acids. In today's class we were going to discuss few sulfur containing amino acids, methionine and cysteine metabolism. So, the concept which will be covered in today's session are related to methionine metabolism, where we will discuss how methionine is activated, how that activated methionine takes part in different types of metabolic activities, then the catabolism of methionine, and finally few clinical disorder related to methionine metabolism. Then we will move on to cysteine, how cysteine is synthesized in our body and how it is degraded, then the metabolic roles of cysteine, and finally few clinical conditions related to defective cysteine metabolism. So, let us move on to methionine metabolism. So, methionine, the amino acid is represented as met or M. This is the code how amino acids are represented. Now, methionine is one essential amino acids, and remember all the essential amino acids, the characteristics is that essential amino acids are not synthesized in our body, those are basically obtained through diet. So, again methionine is not synthesized in our body. It is a sulfur containing amino acid and also glucogenic amino acid. So, glucogenic amino acids somehow contribute to form intermediates which finally gives rise to glucose on requirement. Then the major function of methionine is that it acts as methyl group donor in different types of reactions in our body. So, here you can see the structure of methionine. So, this is our sulfur containing amino acid here. Now, how the activated methionine is formed? So, the activated methionine is SAM, S-adenosyl methionine. So, by the name it is evident that there will be addition of one adenosine molecule to the methionine, and where from that adenosyl group comes? It is from ATP. So, ATP here is the adenosine group, adenosyl group donor, which with the help of adenosyl methionine adenosyl transferase forms S-adenosyl methionine or SAM. And also here you can see the pyrophosphate as well as the inorganic phosphate is released. Now, remember S-adenosyl methionine, here we are getting one high energy methyl group containing molecule SAM. That is why SAM can donate this methyl group to various reaction where methyl group is required with the help of the enzyme methyl transferase. So, once the methyl group is donated from SAM, it forms S-adenosyl homocysteine. S-adenosyl homocysteine then undergoes hydrolysis where adenosine is released. So, what we are getting is homocysteine. Now, homocysteine either it can undergo catabolism or it can reform the methionine. Now, how the methionine is reformed from homocysteine? Via methionine synthase. The enzyme here is methionine synthase, and with the help of the molecule methyl tetrahydrofolate. So, basically methyl tetrahydrofolate here transfer its methyl group to homocysteine. So, we are getting methionine, whereas methyl tetrahydrofolate is converted to tetrahydrofolate, and this reaction is vitamin B12 dependent reaction. So, here you can see methionine while forming methionine, the methyl group donor is methyl tetrahydrofolate. But the active form of methionine, that is S-adenosyl methionine, is another methyl group donor, which is the activated methionine again. So, here you can see that homocysteine can be reutilized to form methionine, or it can undergo catabolism to form propionyl coenzyme A. So, the end product of methionine catabolism is propionyl coenzyme A, and you know the fate of propionyl coenzyme A via methyl malonyl coenzyme A formation. It forms succinyl coenzyme A, and the succinyl coenzyme A can enter TCA cycle. So, that is why methionine is called glucogenic amino acid. Now, how this propionyl coenzyme A is formed? So, from methionine we have seen there is formation of homocysteine. Now, homocysteine forms homoserine and cysteine via reacting with serine, another amino acid. So, here you can see homocysteine and serine reacts to form cystathione with the help of the enzyme cystathione beta synthase. Then cystathionase basically hydrolyze this cystathione molecule to form cysteine and homoserine. So, here you can see cysteine, the amino acid cysteine is actually formed from during the catabolism of methionine. Then homoserine undergoes deamination to form alpha ketobutyrate. Here you can see alpha ketobutyrate. Then alpha ketobutyrate undergoes oxidative decarboxylation with the help of the enzyme dehydrogenase, releasing one molecule of carbon dioxide, forming propionyl coenzyme A. So, this is how propionyl coenzyme A is formed from methionine, and during the process we also get cysteine, the other sulfur containing amino acid. So, this is the process. From methionine we are getting S-adenosyl methionine. S-adenosyl methionine donates its methyl group in via methyl transferase enzyme. What it forms is S-adenosyl homocysteine, which on hydrolysis releasing the adenosine group, it forms the homocysteine. Homocysteine when reacts with serine, it forms cystathionine. Cystathionine then forms cysteine and homoserine. Homoserine undergoes deamination and oxidative decarboxylation to form propionyl coenzyme A, and this propionyl coenzyme A takes part in neoglucogenesis. So, what are the different role of methionine? So, definitely methionine is one glucogenic amino acid as well as the source of cysteine we already have discussed. Apart from that, the major important role of methionine is as a methyl group donor to different types of reaction. Here I have mentioned few important one like formation of epinephrine. So, catecholamines are formed from methionine. One catecholamine epinephrine is formed from methionine. So, here you can see from norepinephrine with the help of the enzyme methyl transferase, and definitely the active methionine, that is S-adenosyl methionine, is the methyl group donor. So, with the help of the methyl transferase there is formation of epinephrine. Then creatine is also formed from with the help of methionine. So, creatine is one nitrogen based compound which is adequately present in the skeletal muscles, and that is formed from the amino acid glycine and arginine. The detail will be discussed in the respective glycine metabolism, but here you can see from glycine and arginine guanidoacetic acid is formed. Guanidoacetic acid takes methyl group once again from S-adenosyl methionine with the help of the enzyme methyl transferase to form creatine. Then choline is also formed from methionine. Here the methyl group is donated thrice. So, from the amino acid serine there is formation of ethanolamine by decarboxylation. Now, this ethanolamine undergoes methylation for 3 times. So, this is one methylation, methyl group donor is SAM. So, here you are getting methyl ethanolamine, another methyl group is added forming dimethyl ethanolamine, then the third methyl group is added to form trimethyl ethanolamine, which is also known as choline. So, choline is also formed with the help of methionine. Finally, melatonin is formed from serotonin via taking the methyl group from SAM. So, the detail of melatonin formation will be discussed in tryptophan metabolism, because melatonin is a derivative of tryptophan. So, here in short you can see from tryptophan there is formation of 5-hydroxytryptophan, then there is formation of serotonin which is also known as 5-hydroxytryptamine. So, this is one amine. Now, 5-hydroxytryptamine undergoes acetylation to form N-acetyl 5-hydroxytryptamine, sometimes we call it N-acetyl serotonin. Then N-acetyl serotonin undergoes methylation to form melatonin. Now, different polyamines are also formed from via methylation with the help of methionine. Now, these polyamines are important because these are the positively charged compound, and those positively charged compound basically binds with negatively charged compound like DNA, like the amphipathic membrane lipids, phospholipids, and they alter their function or they modify or modulate their function. So, these polyamines are also synthesized from with the help of methionine. So, here is the list of different types of transmethylation reaction. Here you can see formation of creatine, N-methyl nicotinamide, catecholamines like epinephrine, metanephrine, normetanephrine, then choline, anserine, melatonin, methyl histidine, methyl lysine, and as well as methylated tRNA are also formed from with the here the methylation reaction occurs where the methyl group donor is S-adenosyl methionine. Then few disorders related to methionine metabolism. Now, one such important disease is homocystinuria. Now, homocystinuria can be genetic or can be acquired. The genetic or the disorders which are arising from mutations are basically of autosomal recessive conditions. Now, there are different types of homocystinuria. The 4 types here we have I have presented via the diagram. So, here you can see homocysteine is basically formed from cystathionine with the help of the enzyme cystathionine synthase. Now, this enzyme if defective it causes type 1 homocystinuria. Next, homocysteine forms methionine with the help of the enzyme methionine synthase, where the methyl group donor is basically methylcobalamin or vitamin B12. Now, this vitamin B12 comes from dietary sources. So, if there is dietary deficiency, that is type 4. Now, remember type 4 homocystinuria is not the genetic variant, rather the acquired pattern where vitamin B12 deficiency occurs from dietary deficiency. Now, this vitamin B12 forms methylcobalamin with the help of the enzyme cobalamin methyl transferase. When this enzyme is defective, it causes type 3 homocystinuria. And where lies the type 2? So, you can see the methyl group donor to form methylcobalamin is methyl tetrahydrofolate. After donating the methyl group to the cobalamin B12, it forms tetrahydrofolate, and from tetrahydrofolate methyl tetrahydrofolate is formed with the help of one reductase enzyme. When this reductase is deficient, it causes type 2 homocystinuria. So, once again to the different types of homocystinuria, type 1 is due to the deficiency of cystathionine synthase, which is directly related to the formation of homocysteine. The other variants of homocystinuria are basically where methionine synthesis from homocysteine is defective. The type 1 is due to the deficiency of the reductase enzyme, which is forming methyl tetrahydrofolate. Type 3 is defect in formation of methylcobalamin, where the defect lies in the enzyme cobalamin methyl transferase. And finally, the type 4 pattern, that is the acquired homocystinuria, which is due to dietary deficiency of vitamin B12. Now, for all the homocystinuриas, the normal homocysteine level is 5 to 15 micromole per liter. The autosomal recessive pattern, the increase is around 50 to 100 times, whereas in case of the acquired pattern or the dietary deficiency, the defect, the homocysteine level is not much raised. Now, what are the manifestation of homocystinuria? Mostly the initial years, like initial 1 to 2 years, the babies remain asymptomatic till around 3 to 5 years, where there is adequate or enough accumulation of homocysteine, or rather enough deficiency of methionine, manifestation of homocystinuria occurs. The initial or the very first detected manifestation of homocystinuria is abnormality in vision due to ectopia lentis. Now, what is ectopia lentis? It is basically the displacement of eye lens to the posterior chamber of eye. For that, there is disorder in vision. Then there is mental retardation, bones are weak due to osteoporosis, and also there is chances of thromboembolism due to coronary artery disease or cerebral strokes. Now, why there is increased risk of thromboembolism due to homocystinuria, we will discuss in further slides. So, what are the basic abnormalities in homocystinuria? So, as we are talking about homocystinuria, it is evident that there will be excretion of homocysteine in urine, as well as what we will get in blood, accumulation of homocysteine. Also, methionine can be increased in blood. Then একটু ব্রেক দিচ্ছি, হ্যাঁ, চোখ কুটকুট করছে। Then we are going to discuss the screening and diagnosis of homocystinuria. Screening is done by cyanide nitroprusside test. So, what is the function of cyanide here? When treated with cyanide, it basically reduces the disulfide bond present in homocysteine to produce homocysteine. Remember, the sulfur containing amino acid cysteine or cysteine here, it contains the sulfhydryl group, free sulfhydryl group. Now, the free sulfhydryl group has the tendency to get oxidized. So, basically what happens when oxidized, it forms the two molecules of cysteine are actually joined to form cysteine. So, here please notice the spelling, it is T-E-I-N-E, whereas cysteine is only T-I-N-E. So, basically cysteine is the diamino acid, whereas cysteine is the single amino acid we are talking here. So, once the disulfide bond is reduced, homocysteine is also forming homocysteine. Now, the cysteine reacts with the nitroprusside and forms a red color. Now, the problem is, if there is cysteine itself, only cysteine, not homocysteine, only cysteine is present in urine, that can be misleading, because homocysteine as well as cysteine, they both will react with nitroprusside. So, how to differentiate? With the help of the test silver nitroprusside test. Here we have talked about cyanide nitroprusside test, and this is silver nitroprusside test where there is no cyanide. So, basically in silver nitroprusside, the sulfhydryl group will not be reduced, whereas cysteines will form red color. So, what will happen? Homocysteine will not react to the silver nitroprusside or will not give rise to red color while treated by silver nitroprusside. So, how to discriminate cysteinuria and homocysteinuria? By doing both the test, both cyanide nitroprusside test as well as silver nitroprusside test. So, what will be the effect? There will be positive cyanide nitroprusside test, but negative silver nitroprusside test in case of homocysteinuria. But remember, this is only screening test. What is the confirmatory test? By first detecting the homocysteine level in plasma and also measuring the activity of cystathionine synthase enzyme. So, these are the confirmatory or diagnostic test for homocysteinuria. So, the defect in congenital or autosomal recessive patterns are related to the different enzyme deficiencies, whereas acquired homocysteinemias or the increased level of homocysteine in blood are mostly due to either nutritional deficiency like cobalamin, folic acid, pyridoxine, which were needed for synthesis of homocysteine or to methionine formation. Then there can be metabolic defects like chronic renal disease or hypothyroidism, and also few drugs like folate antagonist or vitamin B12 antagonist or pyridoxine antagonist. They actually produce a scenario like folate or vitamin B12 or pyridoxine deficiencies. Then estrogen antagonist also causes hyperhomocysteinemia. Nitric acid antagonist, they also causes hyperhomocysteinemia. So, all these vitamins, remember, cobalamin, folic acids or pyridoxine, they were required, if you remember, from homocysteine to methionine formation. All these vitamins were required, and their deficiency will hamper the methionine synthesis from homocysteine, which will give rise to increased homocysteine level in blood, causing hyperhomocysteinemia. Now, remember, hyperhomocysteinemia of maternal pattern can give rise to neural tube defects in fetus. So, very high dose of folic acid is advised in pregnancy. So, this is one important question, why high dose of folic acid is advised in pregnancy, because maternal hyperhomocystinemia can affect fetus causing neural tube defects. Now, why hyperhomocystinemia or increased homocysteine is a risk factor for coronary artery disease? Because homocysteines are considered as prothromboembolic molecules. Now, how the coronary artery disease is propagated via homocysteine? By defective collagen synthesis. Now, how this collagen synthesis is affected by homocysteine? So, I am not going much in detail with the collagen synthesis, but I want to highlight one very important enzyme, that is lysyl oxidase enzyme, which is required for cross-linking and maturation of collagen. So, this lysyl oxidase enzyme required for formation of hydroxylysine, and that hydroxylysine is important for collagen cross-linking. Now, copper here acts as a cofactor for the proper action of lysyl oxidase. Now, this lysyl oxidase enzymatic activity is basically hampered by homocysteine. How? The copper ion forms a complex with homocysteine, and that copper ion is not now available for the action of lysyl oxidase. So, basically what is hampered? Collagen cross linking. Similarly, homocysteine and homocysteine both react with lysyl semialdehyde. So, lysyl semialdehyde is a product of lysine via oxidative deamination. This semialdehyde is also important for formation of collagen fiber cross linking via formation of Schiff's base. So, in both the cases, collagen cross linking as well as collagen maturation is affected. So, what is actually affected? The vascular walls, formation of vascular walls as well as remodeling, that predisposes to thromboembolic risks. Apart from this, homocysteine thiolactone, a free radical generated from homocysteine, also acts as prothrombotic molecule. Basically what it does, this thiolactone thiolates the LDL particle. Now, this thiolated or modified LDLs have the tendency to aggregate and block the vessels, causing coronary artery disease. Next, we will move on to cysteine metabolism. Once again, cysteine is another sulfur containing amino acid which is also glucogenic, but remember cysteine is a non-essential amino acid. So, basically cysteine can be formed in our body, and we have already discussed that cysteine is actually formed from methionine. Now, as I have told you already that cysteine is the diamino acid form of cysteine. Once again, concentrate on the spelling. T-E-I-N-E is the single amino acid, that is single amino acid, whereas cysteine is the diamino acid. So, here you can see cysteine, this is one molecule of cysteine and this is another molecule of cysteine. They both form together cysteine, the diamino acid. So, once treated with the reductase enzyme, the sulfidyl groups are actually reduced to form the cysteine from cysteine. So, basically we have already discussed that cysteine is actually formed from methionine via formation of S-adenosyl methionine. The methyl group donated form is homocysteine. Then homocysteine forms the cystathione during the catabolism to form cysteine. Now, there is one effect which calls cysteine sparing effect. Cysteine imparts one sparing action on methionine. Now, what is that? There is a requirement of total sulfur containing amino acid in our body, which consists of both cysteine requirement as well as methionine requirement. Now, if dietary supply of cysteine is high, it will basically cut down the requirement of methionine. The minimum obligatory methionine requirement is reduced. So, that is called the cysteine sparing effect on methionine. So, we are coming to the metabolic role of cysteine. So, as we have already discussed, glucogenic amino acid it is. So, here you can see this is the very common chart we have continuously discussed throughout the session of amino acid metabolism, that methionine and cysteine can contribute to TCA cycle. Methionine contribute to TCA cycle via formation of succinyl coenzyme A, whereas cysteine forms pyruvate during its catabolism. Pyruvate can be formed from cysteine via two different series of reaction. So, here you can see in this pathway there is transamination to form 3-marcaptopyruvate, which finally forms pyruvate via donating the sulfur as H2S. The other variety which involves the enzyme dioxygenase is the major variety. So, here you can see there is formation of cysteine sulfonate that forms sulfinopyruvate. Finally, pyruvate is formed. So, this is how pyruvate is formed from cysteine. So, once again remember methionine and cysteine, both the sulfur containing amino acid can contribute to TCA cycle, that is why they are glucogenic amino acid. Cysteine forms pyruvate, whereas methionine forms succinyl coenzyme A. Then moving to the next very important function, cysteine is required for formation of glutathione. We already have discussed that glutathione is a the glutathione is a molecule which contains 3 amino acids. Now, this synthesis will be discussed in the glycine metabolism, but remember the 3 amino acid is glutamine. I already have told you. Again I am reminding that the 3 amino acids are glutamic acid, then glycine and our very cysteine. Now, this glutathione is important with regard to multiple functions. The detailed function once again will be discussed in glycine metabolism, but to remember glutathione is required for amino acid absorption. I have already discussed the Meister cycle in digestion and absorption of protein. Then glutathione is important for various reduction related reaction, and via this reduction based reaction there are various enzymes which remains in active reduced state. Then the integrity of RBC membrane is also maintained with the reducing property of glutathione. Also, this reducing property is important for formation of hemoglobin from methylated hemoglobin or methemoglobin. Then detoxification reaction, where glutathione is important for conjugation with the help of the enzyme glutathione S transferase. So, via detoxification glutathione forms soluble products for clearing of these toxins from our body. Next, another important product is taurine, which is formed from cysteine. Remember, taurine was important with formation of bile acids, taurocholic acids or taurochenodeoxycholic acids, both are formed from taurine. Now, this taurine is formed from cysteine. Here you can see cysteine forms cysteic acid, which undergoes decarboxylation to form taurine. Then there are various enzyme catalysis, basically enzymes which contain sulfhydryl groups. They are mostly based on cysteine, and their sulfhydryl groups remains in reduced form with the help of the cysteine molecule. Then we are going to discuss few disorders related to cysteine metabolism. One is cystathioniniuria. Remember, that is cystathioniniuria, not cystinuria. This is cystathioniniuria. So, here we are talking about cystathione excretion in urine. So, once again to remind, cystathione is formed from homocysteine with the enzyme cystathione synthase. Now, cystathione synthase if defective, that causes homocystinuria, because cystathionine is not formed. So, what is accumulated is homocysteine, whereas cystathioniniuria is related to cystathionine accumulation, and that is because cystathionine cannot be degraded to homoserine due to the defect in the enzyme cystathionase. So, cystathioniniuria is related to cystathionase enzyme deficiency. Once again, it is a recessive pattern inheritance manifested as severe mental retardation. What you will get is increased concentration of cystathionine in urine and blood. So, what you need to do? Methionine containing diet should be restricted, and alternatively what should be supplemented? Cysteine, because this degradation is hampered. So, there will be cysteine deficiency as well. So, cysteine will be should be supplemented. Then pyridoxine as well as folic acid, these vitamins should be supplemented in diet. Then we are coming to cystinuria. So, here cysteine will be released or excreted in urine, and the defect is in different transporters of amino acids. Remember, these type of transporters, these amino acid transporters, they transport cysteine as well as few other amino acids like lysine, ornithine or also arginine. So, commonly this condition is known as cysteine lysinuria. Both cysteine and lysine can be detected in urine. So, that is known as cystinuria is also known as cysteine lysinuria. It is also of autosomal recessive pattern. What you will get, because the cysteine lysines are excreted in urine, cysteine crystals will be deposited in kidney, forms calculus as well as crystals will be excreted in urine, and in severe cases these crystals when deposited will cause renal insufficiency. Once again, the screening is cyanide nitroprusside test. If you remember that nitroprusside used to form red color in presence of cysteine, but the specific amino acid urea, what specific amino acid is present in urine, whether that is cysteine or lysine or ornithine or arginine, can be detected by chromatography. So, what is the treatment? Once again, fluid should be taken adequately so that the urinary excretion can be increased, where the excretion of the cysteine as well as the small calculi as well as the small crystal can be increased. Then alkalinization of urine, it causes it basically solubilizes the crystal, and also the penicillamine. Penicillamine forms complexes and causes enhances the release of the crystals of cysteine. Then cystinosis, this is of autosomal recessive variety of disease and also familial disorder. So, in a family these genetic type of disease can be seen in the related members. So, cystinosis is basically accumulation of cysteine crystals in different tissues. Mostly what is affected is lysosome, because the lysosomal transporter, the membrane transporter of lysosome is defective. And which organs are affected? Liver, spleen, bone marrow, they are affected. So, cysteine crystals are accumulated in liver, spleen, bone marrow, as well as these crystals can be detected in white blood cells. What will happen? A generalized amino aciduria can be seen, and those when deposited in kidney can cause renal failure. Once again, the treatment is to increase the urinary output via adequate fluid intake and also alkalinization of urine causing solubilization of the crystal, and also administration of D-penicillamine. So, what are the key points from this session? We have discussed methionine metabolism. In methionine metabolism, what you need to remember is the active form S-adenosyl methionine, which acts as the major methyl group donor. Then cysteine, cysteine is formed from methionine, and also cysteine is the diamino acid, cysteine is the single amino acid here. It acts as a sulfur donor, rather sulfhydryl group here. Then glutathione and taurine are the two important products which are formed from cysteine, and we also have discussed few disorders like homocystinuria, cystinuria and cystinosis. These are my references. Thank you all. See you in the next class.